sorry. When they came down, did they just give you one mic?
There was one microphone and there was one on the pile of boxes at the back. Just to the
Okay,
left.
okay, perfect, thank you. Is it okay to share?
Yeah, sure.
Yeah,
That's fine. We'll work it out. Should we pop it down on here?
Perfect. Thanks a lot. We don't have enough clipboards. Sorry, we don't have enough clipboards. You can check.
This pencil's there.
When I introduce the judges, hello. Do we want to hand over so you can introduce yourselves briefly, or shall I just do the run through?
I think that let's do that. Yes, you're happy. Yes. No, it's good check.
Okay. Good evening, everyone. And welcome. Good evening. Thank you. Welcome to the Cambridge East of England FameLab final. FameLab is the science communication competition that encourages scientists, engineers, technologists, and mathematicians to share the excitement of their research with others. FameLab is run by the Cheltenham Festivals, and we're really pleased to be part of the FameLab International family. And this year we're really pleased to welcome Gizana McLaughlin, the overall FameLab manager, who's come all the way from the Cheltenham Festivals to be with us this evening. So thank you very much for coming. My name is Andrew. I am the head of programs and delivery at the Cambridge Science Centre. We're an independent charity that engages young people with science, technology, engineering and maths. We make sure that everything is hands-on and craft memorable, awe-inspiring, curiosity-creating moments that can be remembered for a lifetime. As such, we're always on the lookout for great examples of science engagement, and I'm very pleased to be here this evening to witness 7 contestants bring some of the very best to the stage. This is the 20th anniversary of the FameLab competition, and the winner this evening will go through to the UK final at the Cheltenham Science Festival in the summer, and with a little bit of luck and skill, through to the international final following that. Each of our competitors this evening submitted a video, and the FameLab team reviewed all of those, I've been told, while eating copious amounts of popcorn. And they're very excited to see these 7 finalists take the stage this evening. Each have 3 minutes, just 3 minutes, to talk about any scientific topic that interests them. Many have taken on their own research to discuss with us this evening. We do have a very big bell to make sure that we keep time. That is Lucy with the very big bell. Don't mess with her. Following the presentations, our wonderful judges will then ask a brief question to the competitor. The competitors are not allowed any presentation slides. They can only have what they're able to carry onto the stage. So we'll see what that brings. Our judges this evening, we have Dr. Vanilla Glynn, Chair of Cambridge Neuroscience Strategic Manager and supports over 850 neuroscientists from across Cambridge. Professor Nicholas Pugh from the Medical Technology Research Centre at Anglia Ruskin University. Craig Brearley, Head of Research Communications at the University of Cambridge. Jacqueline Garget, Research Communications Manager at the University of Cambridge. Vanessa Bismuth, Communications Manager at the Cavendish Laboratory, Department of Physics at the University of Cambridge, and award-winning journalist and broadcaster Chris Mann from BBC Radio Cambridgeshire. They will be watching and listening with their eagle eyes and ears to look at what our competitors have to bring this evening. They will be choosing the winner and the runner-up, but you all get a vote as well. We'll be looking for our audience favourites. You will be able to vote. There will be QR codes at the interval where you'll be able to vote for your favourites. And during the interval, we're very delighted to welcome back last year's winner, Richard Dyer, runner-up and audience winner at the UK FameLab final, who will be entertaining us while the judges go and deliberate and decide what is going to happen, who is going to move forwards. So with all of that said, I think we're ready to go. So we'll kick off with our very first competitor. I'm delighted to welcome to the stage Manar Al-Saif.
When I was a kid and my baby teeth fell out, my mom would get me to bury them in the garden. Has anyone else done that? She probably just wanted me out of the way and maybe she was keen on composting. But I was thrilled by the license to dig. And I used to wonder, can I bury them deep into the Earth's core? The short answer is no. But Earth has a great way of burying stuff not quite into the core, but deep into the mantle. And rather than bothering with trifles like a child me, it does something useful. It recycles its tectonic plates. See, there's two types of plates on Earth. Continental plates, which always get reworked on the surface, and oceanic plates, which are born of the depths, and to the depths they must return. See, the way to make an oceanic plate is to open a mid-ocean ridge. You know, like the thing you see in the middle of the Atlantic on Google Earth? How do you make one of those? Well, you start by pushing two plates apart. Now that there's a gap in the middle, you've lowered the pressure on the mantle underneath. See, the thing is, mantle is hot, but it's under so much pressure, it's compressed into the solid state. And so when you unload it, you remove some of that pressure, and it can expand. So much so that it melts. And now it's a lot lighter than everything around it. And so it can rise, carried by its buoyancy, until it spills out of the ridge and meets the cold surface of the Earth that chills it to solid that forms the bottoms of the oceans. Back beneath the ridge, we said melt is lower density, and so it becomes a self-sustaining low pressure point. So once it gets going, it keeps going. And more melt will keep coming out and spewing out of the ridge and pushing aside whatever just came out before it. And it becomes a growing oceanic plate. So eventually, the stuff that got pushed further away gets cold. So much so that it gets really dense. Remember, it's made of the same material as the mantle directly underneath it, but now it gets denser than the mantle underneath it and ends up sinking back down under its own weight. And that is plate tectonic subduction. Now, it takes a plate around 100 million years to get cold enough to subduct, and that's not long in Earth time. Continental plates, they've been floating around on top of the mantle for billions of years. They don't subduct because they're made of material that's lighter than the mantle, even when it's cold. And billions of years is a whole order of magnitude longer than the entire lifetime of our forever young oceanic plates, which are moved by density and whose burial, now we think, is a major force that drives all tectonic motion on Earth.
Amazing. Thank you, Manar. Slight concerns for anyone who goes gardening in your childhood home and finds your baby teeth. To our judges, a question for Manar.
Why should I care about what you just told me? Why is it relevant to right now?
Well, we're only just starting to understand how Earth works. We still know more about outer space than we know about the inside of the Earth. So, considering how we're thinking about sustainability and how we can live sustainably on Earth, it's a little bit ridiculous that we actually have very little understanding of how our solid planet functions. And the insides and the working of the insides build the crust and the architecture of the crust and how that interacts with the other layers, with the biosphere and the atmosphere. And so we really need to solidify our understanding, not just because of geohazards. I mean, I think it's crazy we still can't predict earthquakes, which is an immediate effect of these kinds of motions. But we're starting to see that there's so much interaction between tectonics and climate, for example, and life. And in fact, we now think that plate tectonics is one of the main reasons we have life on Earth, because nutrients get recycled through this process of burial and rebirth. So it's pertinent to us as life forms for as long as we hope to live on Earth.
You're obviously very enthusiastic about this subject. How did you come to choose this one?
Back when I was 3, I decided that I wanted to study nature, and I didn't really know what that was. And over the years, I learned that, earth science is what I was really fascinated by. A large reason was because I thought I'd get paid to go hiking, which I have. Once got paid to go snorkeling. That was the highlight of my career. But I just find nature fascinating, and I've always wanted to understand landscapes and what makes landscapes. Why is there a mountain where there's a mountain, and why is there an ocean where there's an ocean?
Any questions from the mic? Very good. Thank you very much. Thank you.
No, I mean, it's a great presentation, kind of like you said, you were very, very passionate about it. I'm trying to think what I want to ask you. I think you've kind of answered everything I wanted to ask. Anything from the other judges?
Well, maybe you can tell us why you're passionate about telling the rest of the world about your research.
It's a lot to do with this, with the same answer of why is it pertinent. It's pertinent to all of us, but I think there's so much awe and inspiration to be shared, and especially these days where so much of us suffer from climate anxiety and the doom and gloom of the climate crisis. I think there's a lot of optimism and inspiration to be learnt from just the fundamentals of earth science, of understanding our habitat and our planet. And I think if we do that better, if we tell that story better, it will open the conversation to so much more people, and we'll get closer to just and sustainable transition, because at the moment the conversation is narrow and held amongst too small a type of person. And I'd really like to see more communities and more kinds of people involved in the discussion. But first we need to show them why and where are the questions. I think there's a big misunderstanding in how much we know. So, earth science is a very young science. Plate tectonics was only really posed as a theory in the 60s. And we still know so little, but, I think people assume that things are understood, and they're just not. We need more brains. We need more perspectives.
Amazing. Thank you, Manar. Round of applause for Manar. And let me introduce our next competitor. This is Tasmin Sarkani.
Over the next 3 minutes, 20 billion cells inside your bodies are going to divide. Isn't that amazing? They're going to round up and split exactly down the middle to make 2 exact copies of the original cell. Now, apart from this being pretty spectacular already, it's also quite a difficult thing to do. If I was to take a balloon and say it's going to divide itself, that would be a magic trick. So how on earth are cells doing this, and doing it so reliably that we can stake our own survival on it? Well, to understand this, we need to know how cells change their shape. And this is a question that's relevant not just for cell division, but for all of biology. Our bodies are made of hundreds of different cell types, all with different shapes that allow them to function properly. For example, we have neurons, which are like thin wires to transmit electricity, or skin cells, which are rectangular so that they can form collectively a barrier. But remarkably, there is just one structure that controls all of these different shapes that the cells have. It's called the actin cortex, and it lies directly below the cell membrane. Actin is a cortex that forms filaments, or basically wires, and it congregates in this cortex. Now, people who use headphones will know that wires can sort of get tangled quite easily, but as soon as you use some cable organizers, you can make some pretty cool shapes out of them. You can make a neuron, for example, if you have some nice cable organizers, a nice sort of long string that you can transmit electricity down. Or for most cells, you have a sort of mesh, which, though a bit more messy, is much more flexible and can accommodate any shape that the cell might need to have. Now, we know how cells can have different shapes with these actin formations, but how do they change their shape? For this, we need another protein that generates a force, because we know from physics that you can't change shape unless you have a force. This protein is called myosin. It looks quite similar to actin, but it has these heads on it and can attach itself to an actin cylinder. like this, at either end. Now, the amazing thing about myosin is that it can walk along the actin filament on either side, essentially pulling them together and creating a contractile force. This is, in fact, how muscles work. In muscle cells, actin is arranged in 2 columns, or many columns actually, with myosin in between it. When the myosin contracts, the columns come together, the cell becomes shorter, and so does the muscle, pulling the bone with it and allowing my arm to bend like that. It's pretty cool, I think. But how does this relate back to our dividing cell? Well, when the cell is in its dividing form, actin and myosin is distributed relatively evenly around the surface. But just when it's rounded up, they accumulate along a ring at the center of the cell. If myosin then contracts around this ring, like it did in the muscle, the ring can get smaller and the cell can divide. So the next time that you're struggling with your tangled headphones, or maybe organizing your cables from your computer, just think about those tiny wires in your cells which are literally shaping your life.
Thank you, Tasmin. I'm also very impressed with what you managed to keep inside your bag. Judges, questions for Tasmin?
Hello. Thank you. That was a great presentation. So are you planning on making science communication part of your future career?
Honestly, I would love to. My hobby is stand-up comedy and my job is science, so it seems like I really love talking to people and kind of making fun things out of science. And I also think, personally, I feel like there's a sort of social responsibility of science to really communicate what it's about to people and to be truthful about what we do and don't know to increase the trust in science today.
Thank you.
Do you have a favorite type of cell?
I'm a physicist.
Nothing wrong with that.
So I don't know. I work a lot. The theory that I do is a lot on sort of embryonic cells and skin cells, and I actually think they're pretty cool. They're the ones that I know the most about. Embryonic cells are crazy because they become every cell in the body, so I think there's a lot of things that I don't understand about that and I find very curious about it. Skin cells have the advantage that they're very accessible because you don't have to go inside the body to get them. And they're also super interesting because how they manage to keep these formations from a physics perspective is pretty fascinating.
Okay, so I've kind of got a double question now. As a physicist, what made you deliver a kind of slightly more biological type of FameLab performance, which is really brave, I think, and well done, because I wouldn't, you know.
So I say I'm a physicist, I'm really a biophysicist now, but I trained in physics, so in terms of favorite cell, I would say that I don't have as much of a preference. But my work now is really, the topic of it is very biological. If I was going to do a talk for FameLab, it would be about the biology, because the physics is actually, I would say, too abstract on its own without the biology to anchor it. So that's why I decided to talk about biology. And I ended up in biophysics because I was really interested when I was studying in my undergrad about these collective phenomena. So things where you understand the individual components when you look at them one by one, but as soon as they start to work together, you have phenomena that you couldn't necessarily have predicted with just some of them. And biology is full of these things, and this is a great example.
And my actual question was, what happens, so I loved the fact that you kind of came back to this overarching question, how cells change their shape. So what happens when that goes wrong, when that process goes wrong? Who cares?
I would care, because, well, for example, a very sort of common example is that when you have cells, so basically what I didn't mention because it's not really understood, is that the shape of a cell is very connected to its genetic makeup. So if you have certain genes expressed, it will send signals to the surface. And we don't really know how that happens, that will change its shape. So when you have cancer cells that mutate, it can change the shape of the cell. So what usually happens, so you know when you have a primary tumor, and then the bad thing is when a cell metastasizes or travels from the primary tumor and becomes a secondary one somewhere. To do that, that cell has to become much more floppy so that it can migrate and then reach the bloodstream from the initial one. And that happens because of these mutations. And but the thing is, if you just had the mutations and no shape change, you would probably be okay because it wouldn't be able to travel. But the fact that you have this shape change going wrong is a real problem.
Yeah, okay, that's really interesting, because I guess it's always nice to know what happens when it does go wrong. And any questions? Okay, well done. That was really, really good.
Thank you so much.
Thank you, Jasmine. And now to our third speaker. Let me introduce to the stage Anna Jansen.
Hi folks, can you all see me okay? Wonderful. I really hope so, because you're spending a lot of energy right now just viewing me, my beautiful face, and the face of my wonderful competitors. If you didn't know, your brain is 2% of your body weight, but it uses 25% of your energy input. And your visual system has a lot to do with that. It takes a lot of energy to run your eyes and run the transduction from vision to action on vision. The visual system is made up of two big components, your eyes, too, hopefully, and your optical lobe, which is back here, and is about 20% of the total mass of your brain. Now, if you can't tell, I'm a vision researcher, and I don't do vision work in mouse or humans, but I get to work with the lovely octopus. Now, octopus are a great model for vision because they have a camera-type eye, which is the same type of eye that we have. And we don't see camera-type eyes a lot in animals that are invertebrates, where they don't have a central skeleton. So, really fun, really cool. Now, unfortunately, maybe fortunately, camera-type eye is where it ends in terms of similarity between the octopus and the human visual system. The octopus brain looks a little bit funny. These big lobes here are the optic lobes, and that is the equivalent to your occipital lobe, and they sit directly behind the eyes, and they actually make up 70% of the brain. The central bit here is what we call the central brain. It's donut-shaped because their esophagus runs through it. But most of the neurons are here. So, as you can tell, vision is very important to octopus and their ability to interact with their environment. You may know that octopus are really intelligent, even though they're mollusks, like they're related to clams and snails and stuff. They can solve problems and hold grudges and, basically everything your average toddler can do. But vision is super important as well because they have this additional sensory modality that is camouflage. As a vision researcher, camouflage is fantastic because I can see what they are seeing on their body by the change in their skin patterning. And they use this for everything from avoiding predation to mating to general hunting practices. So I hope that I've shared a little bit with you about how important I think octopuses are to neuroscience research, in particular vision, and encourage you to look inside yourself and look to the animals around you to see similarities and differences.
Thank you, Anna. Our brain might be 2% of my body, but I think long ago it stopped using 25% of my energy. Judges, any questions for Anna?
Yeah, well done.
Thank you.
Have you made a friend with an octopus?
Sorry?
Have you made any friends with octopuses, like the movie?
Oh, so I do molecular biology, so all of my octopus work is actually using their DNA. Okay. And I'm able to extract proteins directly, so I don't do a lot. We can generate a bunch of DNA from one animal. We don't have to use a lot of other information. We don't have to use a lot of other animals for that work in particular. So I don't interact with octopus directly, unfortunately.
Okay.
They hold grudges, so...
Yeah, no, they do. They do. They get mad at specific trainers, especially if they're given food that they don't like from particular people. And they've been known to escape enclosures and put shrimp in people's coffee mugs. So.
Okay. Good to know. I wish you told me that in the presentation. That's really interesting. Tell me why I should care about what you're talking about.
So obviously, I hope all of us have corrected or good vision. And the work that I'm doing is really looking at the fundamental principles of vision and how vision has evolved over time in different versions of things to get at the internal mechanisms and hopefully figure out, okay, this one particular protein is really important for this particular function. We have it in both of these animals. how can we apply that biomedically in the future?
So I was going to ask about evolution and the evolution of your mollusk eye compared to our eye. Is it related, or are they completely different evolutionary pathways for these?
So there's an argument on whether it's convergent or parallel evolution. We have the same key gene that is implicated in vision, but our closest common ancestor between the octopus and the human was about 500 million years ago, and it looks something like a worm and it didn't have eyes. So everything that the octopus has and everything that we have is completely independent in terms of eyes and optic lobes. So.
It's very interesting. I'm going to ask another question as well. So we see in 3 colors, don't we? We see red, green, blue. Mm-hmm. What do octopuses see in?
It's a great question. So they actually only have one photoreceptor, wherein we have a bunch of them, which is how we're able to see color. And we assume that they can see color in some way because of the camouflage. Like they have 3 different pigments that they use to create different maps of things, but scientists have not actually figured that out yet, how they're able to perceive a color or a proxy for a color so that they can actually do the camouflage behavior that they're trying to do. So.
Hi. 2 questions. Did you make that prop yourself?
Sorry?
You make your prop yourself?
Yes, I did. I did knit this myself and the brain. So I'll be posting a pattern for the brain if anybody wants to make one for themselves.
Brilliant. Well done. And why did you get into this research in the first place?
Before I started undergrad, I really wanted to do neuroscience research because I have a lot of family with addiction issues and chronic pain conditions. And when I got into research, I actually started in a very basic science lab that does work in sensory transduction in mice. And when I came here to Cambridge for my master's degree, I was really excited about this octopus project that was on offer. And I mean, octopus, who doesn't want to go work with the octopus, right? And now I'm really excited about vision and evolutionary principles of vision. So
that's great. Yes, you're clearly very enthusiastic about your research. Thank you.
Awesome. Thank you, Anna.
Thank you.
Applause for Anna. And now our 4th competitor. Please welcome to the stage, Spatika.
I love it when people mispronounce my name. Fortunately, that didn't happen today. But honestly, Spatika is a name in a language that barely any people speak. So I don't blame people, I blame their brains. You see, the thing about your brain is that you learn languages in your early childhood, and it stays with you for life, because that's when your brain is fantastic at picking up accents, the right intonations and pronunciations. And we call this a critical period for language learning. It's a point in time when your brain is flexible and adaptable, and it takes on the world and all its impressions. But a critical period also starts to close when you're growing up. So the brain in its critical period is sort of like a piece of clay, and it's picking up these impressions. But as you start getting older, it becomes a lot more hardened and set in its ways. And you're now left with all of these impressions for life, which is why you're now 27, doing a PhD, wondering why you're doing it, and possibly living with all the traumas of your childhood. But what makes this interesting is that we could possibly start to reopen critical periods in an adult brain. Think about it. We could potentially send this brain back into its youthful state, and researchers have just started to do that. A group of people decided to take a bird species called zebra finches, which are songbirds, which, very similar to human beings, learn songs which constitute their language from their parents, and this only happens in their childhood. When they become adults, they're incapable of learning new songs or adding to their vocal repertoire, and that's because these critical periods have closed. So what they did was take these birds and manipulate those brain regions which produce the songs. What they did was specifically silence those neurons or brain cells which are responsible for closing critical periods. And what this did was actually lift up the ban on vocal learning. Adult birds were now capable of adding new songs to their repertoire, and the best part was that it didn't affect what they had learned until then, which meant that it wasn't overwriting anything and was completely new. And this is fantastic because it means one day we could use these techniques in patients with neurodegenerative disorders, with stroke, and with adult aging brains, and potentially give them a window of opportunity and accelerate their recovery by sending them back into a younger state. And let's be honest, as humans, we're only ever interested in increasing our longevity. We're also not just trying to live longer, we're trying to live younger. At least that's what everyone tells me when they say that they peaked in their 20s. Fortunately for me, this is not going to be a problem. I chose to be in academia, where I'm not going to peak until I'm in my 50s. Thank you so much.
Thank you very much, Pratika. Nailed the pronunciation, feeling smug. Questions from the judges?
Thank you. It was really fun. I love that you've touched on so many, like you started with language and then came on to brain and then manipulation of the brain. So what do you study, actually?
Well, I study a part of those things. I study how to manipulate the brain. I do that. But I don't study language. I study motivation because I'm looking at depression. So I do use animal models, and I'm trying to manipulate the brain and see if we can understand these critical periods and what can mess your brain up during development and make it go wrong in adulthood. My whole PhD is literally about saying, can we go back to your youth and address depression rather than waiting for you to become old and sad?
So is it psychology or psychoanalysis, or is it biology?
It's more biology, definitely.
Can I ask a question on behalf of the older people in the room? How did they manipulate the brains of the birds? And second question is, you said there was no negative drawbacks to this. Can you imagine what negative drawbacks would be to sort of reverting a brain to a younger state?
Well, I said it didn't overwrite anything that they had already learned. I imagine there could be negative drawbacks to this, so I would not go so far as to say there aren't any negative drawbacks. But the technique they used is what is called optogenetics, where you literally use a pulse of light to manipulate a neuron. That's a very temporally precise technique because it operates within milliseconds, but it is also very invasive right now, and that's why it's only possible to be done in a non-human model system. So there's a long way to go, and I think what could potentially be problematic if we ever get this down to humans is what people might use this for. I just gave a good example of use that has a benefit, which is helping people recover, but you could use it for any other purpose that could just enhance somebody's performance in areas where you don't really want it being done. Does that make sense?
I was going to ask another question on behalf of the older people here. Are there any kind of tips for how I can revert my brain to a useful stage?
Well, it depends how old you are.
25, thereabouts.
Well, I mean, I know that everybody tells you that people doing crossword puzzles and cognitive activities throughout their lifespan are more likely to, continuously benefit from it as they grow older and remain cognitively able. I don't think you can actually turn it back into how it was when you were 7 or 17.
You probably don't want to.
Yeah, that too.
The impulsivity.
Awesome.
Oh, sorry. No, I'm fine because yeah. You did really well.
Awesome. Yeah, thank you, Pratika.
Thank you. Thank you for pronouncing my name right.
Wonderful. And let's invite to the stage our next competitor. This is David Hardman.
Okay. Could I have your finest drum roll, please? Thank you. Okay, yeah, thank you. I like you already. Thank you. But you don't seem too impressed. But think how impressed you would have been if a robot had just come up here and done that. Because robots that we actually see out there in the world at the moment are designed to do one thing well. And that thing might be putting together cars in factories. It might be vacuuming your living room floor. But they're not designed to be good at general purpose tasks in the same way that humans are good at general purpose tasks. But if they were, we'd have robots that we could deploy into disaster zones, into rescue missions, where it's physically dangerous for humans to go and give help. Or we'd be able to send them into surgeries where they could perform different procedures with precision and improve there. But to do that, they'd have to perform well in the real world. And the real world is noisy, it's messy, it's unpredictable, which as humans we don't mind because we've grown up in the real world and we've learned all its rules. I know intuitively that if I place an object on a surface and don't touch it, that it's not going to go anywhere. But I've also internalized the concept of gravity, and I know that given the chance, that ball would fall to the floor. But robots don't know that. So if I had a robotic hand that I wanted to teach to catch a ball, then sure, I could program it to close and open at just the right times, and it might be able to make some catches. But as soon as there was some noise, a slightly heavier ball than normal, or a gust of wind, then it would fail, because it doesn't have a sensory system to tell it any of that information. So the difference, if I had instead brought along a human hand, would be that it's covered in a skin that tells me where it's being pressed, how hard it's being pressed, how many things, what temperature it is, if it's damaged. It's just me. And it turns out that sensorized skins are a really important part of building general-purpose robots and of augmenting existing prosthetics. And so that's where my research sits, in designing these sensorized skins so that we can teach robots to do more complex tasks. But first, maybe it's catching a tennis ball. Thanks very much.
Amazing. Thank you, David. The robots are coming from us as soon as they learn how to catch balls. Questions from the judges?
How far are we off having human-like robots, do you think?
It's a good question, and the bottleneck at the moment is the bodies of robots. So you will have seen ChatGPT hype emerging, and we're very good at the moment at designing the brains of robots. But the bodies are so far behind. We do struggle for things to catch balls or to walk the length of a room without falling over. So in terms of bodies, we're a few years off, to say the least.
Nice work, David.
Thank you.
Enjoyed it. What are the dangers of teaching robots all this stuff? People worry that they will take over.
Yes.
So will they?
It's a question that should be asked, and no, if we play everything right. So again, it's this difference between brain and body. So I would argue with the bodies of robots, we should improve them, design the best technologies we can in the same way we should have designed smartphones or we should have designed planes and cars. But the brains, it's the dangerous bit when you put a control system into this technology and what it can do there. So that's the bit that should be heavily regulated, is when we apply these brains into these bodies, especially the pace that it's moving at the moment. That interface requires the regulation to stop any takeovers happening.
Well done, David. That was really interesting. A really good use of props, actually. I don't know if my question's going to come out really mumbled, but you said that robots, the world of robots kind of has the brain bit kind of sorted out. But surely the sensory part that you're working on, like, I mean, that is part of the brain. You can't have one without the other.
Yes, you're absolutely right. So the brain is by no means sorted, but it's further ahead than the body is. But all of these will tie together. Yeah,
but that's kind of what I meant. Like when you said you were kind of working on sensory skins and stuff like that, like surely that's quite kind of intrinsically linked with the signaling to the brain.
Yes. Yeah, so you will never be able to design one of these systems by itself. If you design a single component, it's always going to interface with the rest of the system somewhere. So I focus on the technology of the skins and the actual signals that it is able to output that a brain could then take on board, provided those signals are good enough and feed into the rest of the system. But to do that design, you have to consider what that brain is, how it's good, how it's bad, the kind of information that it actually needs to receive.
And are you working alongside then other people who are doing other discrete aspects?
Yes, robotics is hugely multidisciplinary, so you never get someone who can do it all. It's very much a work with people who can fit in just where you leave off, and you can pick up where they've left off.
Okay, thank you very much.
Thank you.
Any other questions?
So what do you think will be the best kind of robot body? Will it be a human body or an animal or something else?
Somewhere in the middle will be the best case of body. So humans and biology and animals are great for us at the moment because evolution has provided billions of years to get things right, not perfect, but working in the physical world, and much better than we could just sit down and design on our first attempt. So nature and bio-inspired designs are an excellent starting point to design what we want, but we don't have exactly the same constraints as nature, for example, in needing to find food to have our energy source. So having something that identically copies nature isn't likely to be the best way, but it's an excellent starting point for our designs.
Great, thank you.
Can I throw in a question?
Please do.
So you see the videos of the Boston Dynamics robots on TikTok, and I'm down with the kids. Brain is still plastic. But they're doing sort of parkour and dancing things. That seems quite human-like and advanced, but you're saying they're such a long way behind. What are they missing despite how advanced and human they look?
Yeah, I'll say they are, of course, very impressive engineering systems, and watching this do a backflip in what could be a curated video at some occasions, but you never see them threading a needle and thread, for example. That dexterity is a really tricky thing for humans as well.
You can watch me do backflips in curated videos as well. Round of applause for David. Thank you very much. And please welcome to the stage our next speaker. This is Mili Ostojic.
So if you're like me and you get really excited by identifying birds when you're walking out and about, you might come across a situation where you see a bird flying in the distance or it's in poor lighting and you can't quite identify it because you can't really see its appearance. And if you've never had that problem, then either you're not very into bird watching or you're way better at it than me. And either way, let me know what that's like. I'd love to know. So what do you do when you're in this situation? How do you identify the bird? Well, one way that I've learned how to do it is using flight pattern. So when birds fly, they use a combination of flapping and gliding motion. So flapping or just wings out gliding. And the amount that they use these two flight modes depends on a lot of factors, but in particular size. Flapping is a very energy-intensive mode of flying. So smaller birds will tend to flap their wings for short periods of time and then glide for short periods of time, whereas larger birds will tend to glide most of the time because they need those big energy savings much more than smaller birds do because it takes them more effort to flap their wings. Wing shape is also a factor. So, for example, ducks have long pointed wings to help them flap their wings continuously, whereas hawks have very large and broad wings to take advantage of updrafts. And so these flapping and gliding motions, they make up some high-level flight patterns that a lot of different species share. So, for example, both ducks and gulls will flap their wings continuously. But we can narrow it down to species groups by looking at the ratio of flapping to gliding. So, for example, both sparrowhawks and swallows will flap their wings for some period of time and then glide, but sparrowhawks will spend more time gliding than flapping compared to swallows. So we can narrow it down like that. But I hear you ask, can we narrow it down even further? Well, yes, we can, because bird flight also differs on the basis of individual flaps of the wings. So when a bird flaps its wings, I'm going to do a little mime, it brings its wings in as it brings them up, which is an upstroke, and then it stretches them out and brings them down in a downstroke. In a downstroke, it's generating lift, And then the upstroke, it's conserving that lift by reducing its surface area to reduce air resistance. And the amount that the bird will use an upstroke versus a downstroke, or the amount of time it will spend in each, also depends on factors like size and wing shape. So we can narrow down the species even further. So we've established that you can use flight pattern to identify the species, but you might be thinking that some of these details seem a bit too detailed. Like, how can you, as a person, take a look at a bird and say, yeah, that's doing an upstroke for 0.1 seconds? Well, maybe you can't, but a computer can. So in my research, I created a program that can take a video of a bird in flight and identify the species from the flight pattern. So it takes the video, splits it into its frames. It identifies whether there's a bird in each of the frames using an AI model and object tracking algorithm. And then in each of those bird frames, it identifies whether it's using an upstroke or a downstroke and builds a flight pattern from that. And from the features of that flight pattern, like the flapping to gliding ratio or the upstroke-downstroke time, it can identify the species. But in creating this program, I did sort of inadvertently teach myself to do that too. So now my brain automatically identifies things. Thank you.
Thank you, Millie. So, computers, bird watching. As David cracks the bodies, we've just created robot bird watchers. That's how it all ends, folks. Judges, any questions?
Yes. If you're not a bird watcher, why is it interesting?
Well, one of the applications that this was initially intended for was for wind turbines. So a lot of wind turbines have systems in place to identify a bird before it comes near the turbine, so they can like switch off the turbines if it's a bird that's like at risk. So they're not going to stop an entire turbine farm for a pigeon, for example, unfortunately. Would love it if they did. But they will stop it for like a hawk or something endangered. And one of the problems they have is, obviously, if it's quite far away, you can't really tell the appearance from that. So it's useful to have these tools using like flight pattern and that sort of thing to actually enhance the identification so that we can better identify these things. And it's also just interesting from like a hobbyist perspective, but that was the main purpose of it, yeah.
I've got an odd question. Have you looked at penguins at all and they're flapping under the water?
I haven't. That would be an interesting one, I think. So far, I sort of took 4 species that have like quite different flight patterns as like a proof of concept. So I haven't really gone in depth on like flightless birds and stuff like that. But I think that would be interesting to see, because obviously they don't flap their wings in the same way. They use them more like flippers. So it would be different for sure, but I think it could be done.
Interesting, Minnie. Enjoyed that. Do the birds have that capacity to tell each other apart? Do they know what the different species and so on? Do they identify all this?
I don't actually know. I mean, by flight pattern, I'm not sure. It's possible they do. Birds are actually like surprisingly intelligent. They've actually got, even though their brains are very small, they have a really high density of neurons. So we don't really know exactly how much they can tell about things. But it's very possible that they would be able to tell these things because they're very, very intelligent. So they probably identify each other through like appearance, song, but it is very possible that they also see another bird flying and think, yeah, that's a bird of prey, for example, because it's gliding, it's hunting, and then they run away if they're a prey. Yeah.
I have 2 questions. One, how quickly can you turn a wind turbine off?
Not very quickly, which is why they have these systems in place that can identify a bird as soon as it enters the sort of airspace of the turbine farm. And then they've also got systems that they're developing so they can actually track the trajectory of the bird, so they know which turbines to switch off in succession to stop the bird from like hitting them. And they don't know for certain, obviously, every single time that it's going to follow the trajectory they're predicting, but it's better than just like leaving it on and hoping it doesn't hit one of them, yeah.
Interesting. And the other question I had is there a plan to roll this out for like birds in general?
Yeah, so I'm in the process of publishing a paper for it, and it is something that I think could be turned into like an app for hobbyists and potentially used in these like commercial settings, but it's not something that I have massively put much effort into because this was my master's project and I'm doing something else now. But it is something that I would be interested in doing. I think it would be useful as like a separate tool to existing apps where you can identify from appearance or from birdsong. I think it would be a good
like
addition.
The app that you identify
Merlin.
your plants from and stuff.
Oh yeah, iNaturalist. Yeah, there is one for birds as well. It's called Merlin. So if you want to identify birds, Merlin is the app. They should stop paying me for advertising them. The amount of times I've mentioned them. Yeah.
Okay, well done.
Thank you.
Thank you very much, Millie. And now we move to our final speaker for the evening. I'd like to introduce through the wonders of modern technology, because unfortunately she wasn't able to be here this evening. Oh, there we go. Let me introduce Lucy Rycroft-Smith.
Thank you so much. Can you hear me all right? I'm assuming that's a yes. I'll just start. Great. Hello, everybody. My name is Lucy, and I'm a mathematician and also an artist, which you'll find out in a minute. And I'm interested in the question, can animals count? And if they could, how would we know? Because there's a particular problem with this, as I'm sure you can imagine, and it's language. We can't just ask them, hey guys, can you count? Are you good at maths? And so it's a bit like if you have a human in front of you and you wanted to find out if they could count, but you couldn't speak the same language as them. One thing you could do is to offer them different amounts of something and see what they did. So very simple example. And with humans, you know, obviously we're talking things like biscuits. We've got some assumptions here. Humans like biscuits, and also more biscuits are good. So imagine if you had a plate of 5 biscuits and a plate of 3 biscuits and you offered them to a human, you wouldn't necessarily need any language. You could observe their behavior, and most likely they tend to choose the most number of biscuits. But there is a problem with that, and I've got some props here that I've made. So imagine your 5 biscuits and your 3 biscuits look like this. Obvious issue here. We've got some confounding variables. So some things here which aren't just about the number of biscuits, because turns out in the human community, types of biscuits are really important. So we've also got an issue here with size. Humans actually might be choosing here because of the size, something to do with area, maybe volume, maybe even length, we're not sure. So if you take monkeys, you can do the same sort of experiment. You can offer them 5 of something, you can offer them 3 of something, and just see what they do over time. And as humans have biscuits, of course, monkeys have, that's right, lemons. If you offer monkeys 5 lemons or 3 lemons, they will very stably over time take the 5, which tells you something, but it doesn't tell you if they can count yet, potentially, because again, we might have these confounding variables. Are they looking at something that isn't just the number of things in the set, the numerosity? Are they looking at the overall area or volume or something else that's important to monkeys that we don't know? So a really cool experiment was done with monkeys, where what they did is they took some monkeys and they put a stage in front of them. So there's an audience of monkeys, and they put some lemons on the stage, so 3 lemons on the stage. Then they raised a screen, and they very, very obviously, these researchers added another lemon, and then they put the screen down. So if the monkeys could count, they would expect to see how many lemons? That's right, 4 lemons. You guys can count as well as monkeys, because they did. When they saw 4 lemons, the monkeys weren't surprised. They looked away very quickly. They went about their monkey business. But when the researchers changed the conditions, i.e. they snuck some of the lemons away, or they put more in, so there were more lemons behind the screen than the monkeys were expecting, the monkeys were shocked. They looked for a long time. There was a lot more going on there. But still, does that mean they can count? Maybe something is going on in the monkey's brain that's a bit like this, maybe this. But here's the cool part. Second part of the experiment, what the researchers did is this time they started with 4 lemons that were medium-sized, but they sneakily swapped them either for 4 huge lemons or 8 tiny lemons. And it turns out when the monkey saw the 8 tiny lemons, they were very surprised. But when the monkey saw 4 huge lemons, they were not surprised. So this proves that the monkeys were counting the numerosity of the set, and they were not looking at other variables. This talk was brought to you today by mathematics, by monkeys, and by lemons. Thank you.
Amazing. Thank you, Lucy.
Thank you.
I do hate unexpected lemons. Okay, can we have questions from the judges, please?
I'm just going to make a little quick comment just because I have the microphone. I think long ago I used to give this lecture about cognition in animals. It was a long time ago. But I remember there was a horse called Clever Hans back in like the either 1800s or 1900s. And they thought he could do maths and count and everything like that. So I wonder, the question I actually have is, I can't seem to get past the whole, do they not just see, whether they, I'm looking at a stage here and there are 6 boxes on it, but like, there's just a load of boxes and it's the size of the overall kind of area. Right. How can you really distinguish between the fact that there are 6 discrete boxes, or there's just a big mush of boxes?
It's a great question. And truly, this is one of the biggest problems in maths. This idea that we think we might be counting discrete things versus actually we're looking at continuous things. And in humans, as in animals, we kind of use them as proxies for each other. So right now, when you're looking at those 6 boxes, yeah, you're not really counting them necessarily. You're doing a little bit there of heuristics where you're saying, well, they're all the same size. I've got what looks like 6, there's 3 in a block and there's 2 of them. So there isn't actually an easy way to distinguish. And I have simplified for this talk. We don't know 100% for sure that the monkeys are counting discrete sets. It just seems like it because of the way that they have tried to eliminate other variables in that particular instance. But it's a great question and it's absolutely fascinating. And ultimately what it comes down to is there isn't actually that much of a difference between discrete things and continuous things for humans and maybe for animals. They're kind of very similar, and at different scales, they become the same thing for us, which is kind of mind-blowing for me.
Enjoyed it, Lucy. Well done.
Thank you.
Surely, if animals needed to count, then they would be able to. Evolution, nature would make sure that if it was necessary for their survival, they could count, or they wouldn't be around.
Well, yeah. I mean, if you think about fish, they need to decide which shoal to go with in a pinch very fast. They need to be able to compare sets. I mean, comparing sets is the most obvious one. Also, things like lions in a pride, they can do a cool thing where they compare things that they hear with things that they see, because they need to do things like if there's an incoming threat, they need to listen for how many roars versus how many other lionesses they can see. I should say lionesses because it is lionesses. And so they can compare sets that way. So yeah, set comparison is pretty important for, survival of almost any species.
Why do you want to know if animals can count?
Well, I mean, I want to know if almost anything can count because I'm obsessed with counting. I'm a mathematician who loves every single thing about maths, but most especially there's a place in my heart for the type of maths that people consider simple and trivial. And counting is it, right? Counting is the thing that everyone thinks, oh, it's just reciting words. It's so complicated. And, I've raised 2 children. I've watched them as babies learn how to count. It's so difficult. It's so interesting. I don't think I'll ever get over the interesting experiments that you can do on all kinds of different species to sort of try and get underneath what's going on in the brain when we're counting. And we just don't know that much about it still. It's absolutely fascinating.
Can I ask a question about us as humans? How old are we when we start to count, and what experiments can you carry out on your children ethically to work out kind of when they can count?
That you can, the short answer is, and this is why I'm saying I'm not a biologist, I'm a mathematician, but the short answer is you can do experiments on your children at any time, including from when they're in utero. So there's a really interesting thing that happens around about 6 months for babies. And it actually was really interesting because David alluded to it in his talk. It's the object permanence thing. So he's saying if you leave a thing there, you expect it to stay there. Well, babies don't get that till I think it's about 6 months. So there is some stuff around counting that you tend to wait till they're about that age, but you can do a lot of experiments on infants. Exactly the thing that we just talked about with the monkeys. You can put some things behind a screen. You can kind of add or subtract really obviously and then remove the screen. And it's the same experiment. Infants' eyes go really big when they're really, really shocked. And they also have a startle reflex, which is kind of like a little frog, which is adorable. So you can do all these sorts of things on infants too. And then, of course, as they get a bit more sophisticated, they can start to use language, they can manipulate objects. And so the kind of idea of, comparing human infants and other types of animals in the way that their brains work around numerosities is so interesting because it is basically very straightforwardly the same kind of thing. There's a heuristic system, and then at some point we start to do the counting, and the heuristic system kind of goes up to about 4. So we tend to be able to kind of go, well, there's nothing, there's 1, there's 2, there's 3, there's 4. In this way that we have called subitizing, where we just know, like when you look at dots on a die. And then after 4, it's a bit more complicated. You need to do a bit more counting. And so we can sort of trace that again in languages in humans, and you can look at the way that different languages have developed different words for number, which also, is a part of this. So you're going to have to stop me talking, by the way, because I will just carry on.
It's fascinating. We don't want to stop you talking. And I'm slightly intrigued by this horse that's been alluded to.
Oh, so let me tell you about clever hands.
Yeah.
Not so
they
clever.
thought it could count, but it couldn't. How did this fool us, this horse?
Well, it was incredibly interesting, more of a psychology thing than a maths thing, but actually not really. So there was this horse that was kind of parading around the country saying, he's incredible. He can do not just knows numbers, but can do sums, very complicated sums. And I mean complicated sums. You know, this isn't monkey stuff. This is, you know, square roots and compute things that can only be done on a calculator. And really a lot of people were fooled for a lot of time. And, you know, the language issue was obviously an issue there. Hans could not speak, just to be clear. This was not a speaking horse. Used to tap things out with a hoof. And so what actually was happening, and it took quite a long time to get to the bottom of this, because it was happening with different people. It wasn't just the one handler that Clever Hans had. When people knew the answer to the mathematical sum, they were giving some imperceptible cues that then told Hans the answer. Imperceptible perhaps to the naked eye, but Hans could tell quite easily. Horses are super sensitive animals, as I'm sure you know, especially to things like movement. And so it was just picking up cues from the humans around it, ultimately. And the reason they found that out is when the humans around it didn't know the answer to the maths. Guess what? Clever Hans, not so clever.
Amazing. So as the human got more excited because it thought the horse was right, the horse just stopped counting.
Basically that.
Job done.
Basically that, yeah.
Amazing. Brilliant. Well, thank you, Lucy.
Thank you so much.
Okay, we now move to the interval. It's time for our judges to retire and do some deliberating following those amazing 7 speakers. But for us, we get to welcome the amazing Richard Dyer back to the stage for this year, the winner of last year's East of England FameLab, who is going to be entertaining us. I don't know how, but we're all about to find out. So judges, if you'd like to retire, and we can give a warm welcome to Richard.
Thanks very much. Thank you. Thanks. Good luck. No, you're okay. Good. Okay. Hello, everyone. I have a question. What do pigeons have to do with the origins of the universe? Well, it's something to do with telescopes, which is convenient because I'm an astrophysicist here in Cambridge. I kind of rely on telescopes to do my job. And there's one telescope in particular, this one here, that is very interesting. And I want to talk to you about this particular telescope because there's an interesting trend in telescopes that starts with this one. This is a very big telescope. It looks at a very narrow region of the sky, and it's monumentally important. What do you think we call such an important telescope? Well, it's called the Very Large Telescope. You think maybe they could have given it a slightly more imaginative name, but that's fine. So moving on, we've got this next telescope here called the ELT. Now, all I'm going to tell you about the ELT is that it's bigger than the VLT. It is indeed the extremely large telescope. Again, could have come up with a different name, but it's fine. That brings me to the third telescope. Well, hopefully. Here we go, the owl. Now, I'll tell you 2 things about the owl. It could be called the OLT, and it's also bigger than the ELT. Any ideas about owl? Any ideas? Overwhelmingly. The overwhelmingly large telescope. Great name. Great name. Now, I joke about these telescopes, but of course they are very important. The VLT, so I should say that OWL was never actually was never actually made. It was cancelled in the end. Probably the clue was in the name, but the VLT is operational. It does some very important things like this here. If you point the VLT to the center of our galaxy, the Milky Way, you see this. Stars orbiting some central point. Now, we can't see what's there exactly. But there's obviously something at the center. The stars orbit around something very small and very heavy, and you just got a hint about what that thing is. It's this. A black hole. A supermassive black hole called Sagittarius A star. And if you take all the telescopes in the world, you combine them together, you can see this black hole. Okay, we might have to do this a bit ad-lib, but yes, the black hole. Now, the interesting thing about a black hole is that it's a hole that's black. And it got me thinking that this trend with names of telescopes extends a bit wider to more astrophysical concepts. And so I want to play a little game with you all today that I like to call the unimaginative name game. And so this is a bit interactive, so feel free to join in. We're going to go through some astrophysical objects, and I'm going to give you a description of them. And all I want you to do is tell me what they're called. And I know you're thinking this maybe sounds a bit complicated, a bit scary. Don't worry. It's an incredibly simple game once you understand it. So I'll give you the first example here. This is a star. It's both big and red. You could say it's a giant star. Any ideas what this star might be called? You're going to be so good at this game. Okay, moving on. This is a mysterious substance that permeates the universe, but it doesn't emit any light. It doesn't interact with light. So it's a type of matter that's dark. Any ideas? Yeah, okay. So I don't mean to, yeah. You're getting the hang of this now, okay. We'll do a slightly more complicated one. This is something that Einstein came up with about 100 years ago. It's a combination of space and time. Space-time. Sometimes just put the words together. Sometimes you don't even need a space between them. Just mash them into a single word. Finally, this is a type of planet that lives outside the solar system. It's a type of exoplanet that's very like Jupiter because it's very big. And it's also close to the host star, which makes it very hot. So it's like Jupiter, but hot. Any ideas? Give yourselves a round of applause. Oh my gosh, wow. You must be astrophysicists. This is amazing. Now, I know what you're thinking, is this entire talk just going to be me naming things? Really obvious names. Well, there is a slightly more, I guess, important point to make here, which is that when we look at things like Jupiter or black holes or red giants or any of these wonderful things that we see in space, sometimes we can feel a bit disconnected. Like these things are somehow separate from us. Like this lives in a different universe to us, but of course we're all part of the same universe. These things are fundamentally real, and I think it's quite nice sometimes that we can be connected to these via the fact that they have silly names and they're discovered by silly telescope names. But it's not really silly, it's kind of profound in a sense. What it's really telling us is that we as humans are connected to the rest of the universe. And I think there's something really nice about that, because it's easy to feel disconnected sometimes from science. And this can become dangerous when we start to feel like scientists are, they have a different agenda to us, or maybe they're gatekeeping, or maybe scientists are weird. Maybe that final one is true, but the point still stands. And I think another way that this is expressed is through things like science communication. Doing science communication connects the scientific world to the real world. But of course, there is no distinction. The real world is the scientific world. And so anything that brings us closer to that world, to these concepts up here, these ideas, these notions, these things that seem beyond us, anything that connects us to them is really nice. And I think perhaps another way to do this is through stories. Because the way that we're taught science is this very linear thread, where one thing happens and then the next thing happens, there's a hypothesis, then it's tested, and then it works, and then we learn something new. But of course, science is much more messy than this. And things go wrong, sometimes people don't know what to expect. There's all kinds of twists and turns. And I think it's really nice to be reminded that science is not really a linear process. The scientific method can take lots of different forms in lots of different ways. And actually, we're all connected to the scientific method in a sense because we're doing it every day. And I think one of the nicest ways to see this is with this particular story here. So this is the Horn antenna at Bell Labs back in the middle of the 20th century. And originally, it was used for communications. And what would happen is you'd send these big balloons up into the air, and microwaves would bounce off the balloons and be used to communicate messages around the world. And then an antenna like this one would pick up those microwaves. But these two scientists here, Arno Penzias and Robert Wilson, had a different idea for this telescope. They didn't want to use it to study terrestrial signals, but extraterrestrial signals, things from space. And so they set out on this mission to see if they could detect microwaves in the rest of the universe. Now, microwaves are the invisible parts of the spectrum. So you have visible light that we can see, and then as you go to longer wavelengths, you get to the invisible part, so things like radio waves, microwaves. And so they were interested in this invisible part of the spectrum, the microwaves. And so they did everything that they should reasonably do to make sure they could detect these properly. They cooled the telescope down. They made it very cold, 4 degrees above absolute zero. And they removed any sources of possible interference with the telescope. And then finally, they were ready to point it at the sky and see what they could detect. What they found was this mysterious hissing sound, this white noise, about 100 times louder than anything you would reasonably expect coming from the telescope. This was very strange. So they double-checked all of their equipment. They made sure that there was nothing obvious, no sources of interference, things that might potentially cause this source of noise. And then they decided to look inside the telescope. And there they saw some pigeons. That was the source of the noise. It was pigeons in the telescope. So they cleaned out the pigeons, they removed any mess, they moved the pigeons out, and then they were finally ready to do their experiments, do their observations. But then the pigeons went back in the telescope. So they had to take them out again. Spoilers. So they took the pigeons out again, and then they were finally ready to observe the universe. And I think this is nice because sometimes in physics things go smoothly, and you find something interesting, like a source of noise in your detector, and you follow it, and sometimes it comes out with something really exciting, it's something new. And sometimes it's just pigeons, and that's kind of okay. Except, when they turned the telescope back on, the source of noise was still there. This hissing sound, this source of noise, 100 times louder than anything you would expect, remained in the data. And it was there day and night, throughout all the various seasons. It stayed there. It was as though the entire universe was bathed in this soft glow of microwaves. Incidentally, 60 kilometers down the road at Princeton University, a team led by Robert Dickey were set out to investigate this precise source of microwaves. The reason being, at the time, there were 2 models of the nature of the universe. There was the steady state model, which said that the universe had been around forever. It didn't have a beginning, it didn't have an end, it had just always existed. But there was another theory that said that the universe had a beginning. And in this moment of creation, lots of matter was produced, but also lots of radiation. As it turns out, that radiation wouldn't be able to travel very far because there would be loads of particles in the way, subatomic particles. The light would travel for a while, then bounce off a particle and go in a different direction. The bounce of another particle, go in a different direction. So if you were living in the universe during this window of time at the very beginning, it would just be a hazy orange. But then, about 380,000 years after that moment of creation, things would clear up. Atoms would be able to form, and the light would be able to travel freely. And it would be able to travel throughout the universe, getting stretched in the process with the expansion of space until that light traveled through the visible spectrum into the invisible part, where we have radio waves and microwaves. When Penzias heard about this, he called up Robert Dickey immediately. He came over, and at once they realized what they had found. It wasn't pigeons, it was the origins of the universe. Something called the cosmic microwave background. One of the most important discoveries we've ever made. This tells us about everything that came afterwards. This moment of creation gives us everything that we see now. This is a profoundly important discovery. Kind of amazing. And it all started just from following this curious source of noise. What first seemed like pigeons was actually the Big Bang. Incidentally, another great name. So we have this scenario, pigeons versus the entire universe. It could have just been pigeons. It could have just been pigeons. But it turns out these 2 unassuming astrophysicists who would go on to win the Nobel Prize for their discovery, had found something truly profound. And I think there's potentially two important messages here. The first is obviously the cosmic microwave background, one of the most important discoveries we've ever made. This tells us about our place in the universe. But then there's also the pigeons, and that tells us about our place on Earth. Because sometimes science is messy. It's a fundamentally, intrinsically human process. And I think that's the wonderful thing about it. And that's how pigeons are connected to the origins of the universe. Thank you.
Amazing. Thank you very much, Richard. Although I do know someone who can develop a system to spot a pigeon half a mile away and turn your telescope off. The Cambridge Festival team will now be handing around QR codes. This is your opportunity. Scan the QR code and you can vote for your favorite of the 7 contestants. As a reminder, that was Manana Al-Saif, Tasmin Sughani, Anna Jansen Spitika, David Hardman, Millie Ostojic, and Lucy Rycroft-Smith. We'll be expecting our judges back in about 5 to 10 minutes. So take this opportunity, scan the code, and vote for your favorites. Thank you very much. My phone in the green room.
Sorry?
I left my phone in the green room.
Oh. I can vote how many times I want to.
You just like...
Have you voted? Know more about the Captain horse as well. The Captain horse would win, in my opinion.
Yeah.
Have you voted?
Yeah, I did.
Voted?
Yeah. No, he hasn't, you know. You haven't seen it on your phone.
I've not voted many times. It's not, you don't want to, you feel it's
I feel, yeah, it doesn't seem right. So.
Everyone is operating the drive in your opinions to the point, to the T.
Thank you very much. I'll try.
Do you need anything? Are you okay?
I'm good, thank you.
Okay.
Self-sorted with some... Let's go around together.
That's a good question. Me? I don't know. People are still voting, so I'm looking for contestants. Actually, I'm gonna get the microphone off Richard. I'm looking for Tasmin. Oh, there she is. Okay, perfect. I have everyone. Just want to make sure everyone is here. But when we announce.
I won't tell you what number that is.
Because it's taking too long. There's nothing for us to do. Be careful with this bell. I'm sitting on the other side.
Yeah, it doesn't make it look like you're really putting a huge amount of effort when you walk around with your glasses of wine.
Really
Do
Last
they?
day. We are.
May I ask what's coming up next? So the chair of the judges will be coming back out and they'll do the announcements of the winners and the runner-up. There'll be an announcement of the audience, the votes, and that's the end of the proceedings. We're in the game. Or is that something else?
I know.
I thought this would end at 10:15, but not at 10:15 now. We're head off.
Quarter to 10.
Will the result of the winner be on the website? I don't know. That's the question.
Sorry, could you repeat the question?
Will the winner of tonight be on the website?
Yes, and we'll announce it, and this is being recorded, so the event in the union will be put on the YouTube channel of the festival, so there'll be a chance. You'll get an email if you're subscribed to Festival Inbox. They will let you know, but it's
if I am on
the festival inbox. Yes, so there will be. Okay, thank you.
Thank you for coming. Have a good evening. Okay.
It's amazing. Are you really cold, Andrew? Are you really cold? Are you really cold?
That's this radio.
Okay. I'm about to go on holiday. Don't you bring in your...
Oh, do I have a cold?
Do I have a cold? That, yes.
No,
Are you really
no.
cold? I've survived 10,000 children, 10,000 people. Oh my God, it's the worst. It was fun. I volunteered myself too. But I was in the family we can understand. I was at Cambridge United Foundation goal, which I thought was right because I love football.
As I remember, yeah.
But funny enough, children also love football.
They were just pelting you with balls.
It was fun.
So yeah, here they come.
Okay, ladies and gentlemen, please put your hands together. The judges return to the room. It is about time to reveal the winner of FameLab East of England Final 2025. And we welcome to the stage Dr. Dervilla Glynn, who's the chair of the judges, who will do the announcements.
Hi, everyone. Thank you so much to all the audience for joining us on what has been another amazing evening of FameLab. I was chatting to Chris earlier in the evening, and we have spent many an evening as judges on FameLab over the years, and we really have met some of the most incredible science communicators, and tonight was no exception. So I really enjoyed myself. I think we all did. We had so many judges, so it made our task even harder. And the nice thing about all the judges tonight, we all came from different subject backgrounds. So there was kind of no real bias. So that's really important. So I'm just going to make a few general comments before I lose my breath about, I guess, the importance of science communication and really what shone out for us tonight. So those of you who work in communication, whether it be in academia, in industry, in the media, I mean, the importance of science communication is ever growing. And you have to remember, a lot of our tax money goes into funding different science. So really it is jobs, it is the absolute job of a scientist to be a good communicator. And it's wonderful that our early career researchers are taking these opportunities. And I don't know about you, but I think it's really brave to stand up here, put yourself forward, and talk about something in 3 minutes, especially if it's not your subject area. Like, I couldn't believe that tonight when some of them really weren't talking about their subject area. I think one thing that all of the judges commented on tonight was how excellent all of our candidates were at questions. So even in the candidates that weren't necessarily up in the top 2 or top 3, I think all of our candidates really relaxed into it when we started asking them questions. It wasn't so kind of prepped and kind of, I guess, you know, stylized or whatever. And they became natural communications. And I think that's really important and really vital going forward. So, dun, 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 dun. We have an audience winner, and then we have a judges winner, and we have a runner-up. So, what do you want first? Do you want the audience winner? Do you want? Oh, we'll do the runner-up first. Oh, okay, well, let's hype it up. So, after much deliberation, and I really mean much deliberation, our runner-up is David. Oh, sorry. All right, okay. Well done, David. Really, congratulations. Everyone really loved your presentation, the fun, the really excellent use of props. So a lot of times, candidates might use props, and you're kind of thinking, well, why do they bother having that? But actually, yeah, except that the hand with the skin, I mean. So, for our judges winner and our audience winner, it is in fact the same person. I know. So this candidate, all of the judges commented on the natural communication style of this candidate. How they delivered a smooth, calm, funny, and professional I guess, performance. One commented on how they could see this person as a professional kind of science communicator or broadcaster in the future. They also liked, we also liked how they chose a tricky subject and made it really accessible. And that can sometimes be a strong ask, and specifically when it's not necessarily their subject area as well. So without further nonsense, our audience winner and our judges winner is Spatica. And with that, it only remains for me to thank my fellow judges. Wonderful to work with you. Thanks.
Awesome. What an evening it has been. I think we need a final round of applause to all of the competitors this evening. So that's Manar Al-Saif, Tasmin Sarkane, Anna Johnson, Millie Ostojic, and Lucy Rycroft-Smith. Round of applause for them. Congratulations, David Hardman, as our runner-up. And to both judges and audience vote, Spatica. Amazing evening. Thank you so much, everyone, for coming to FameLab 2025. We'll see you all next year.